This is Cade. This is our protein synthesis project. We'll first start off with transcription, which occurs in the nucleus. A gene is taken from the DNA, and in the process of initiation, RNA polymerase attaches to the template strand.
And then through the process of elongation, RNA polymerase moves down the template strand and creates a copy called mRNA. And then through the process of termination, the DNA zips back together, RNA polymerase leaves, and you're left with mRNA.
And then after the process of termination, introns are taken out of the mRNA strand, and after what is left of the mRNA strand, this exits the nucleus into the cytoplasm, where mRNA can go through the process of translation.
After the process of transcription, the mRNA that is released into the nucleus and into the cytoplasm goes through the process of translation. Through the process of translation, we have a ribosome, which is composed of a large subunit and a small subunit, which the mRNA runs through. Then we have tRNA, also known as transfer RNA, in which tRNA is composed of an amino acid and the anticodon. As tRNA enters the ribosome, the anticodon from tRNA and the codon from mRNA attach to each other. And as it enters the P site, the anticodon from tRNA and the codon from mRNA attach to each other. And the three letters which compose the codon release the one amino acid in the P site.
So during the P site, as the amino acid is released, this creates the polypeptide chain, which is also known as the protein. And during the E site, the anticodon from the tRNA and the codon from the mRNA detach from each other, which results in the tRNA exiting the ribosome to go find another amino acid.
And this process of detaching an amino acid from the tRNA and the tRNA leaving the ribosome occurs over and over again as the mRNA moves through the ribosome and the polypeptide chain keeps getting built up as more amino acids are added.
Alright, so today we'll be explaining the difference between DNA and RNA. So DNA, it only occurs in the nucleus, it is double-stranded. RNA, single-stranded, occurs in the nucleus and the cytoplasm. The difference between DNA and RNA is that the purines are the same, but the pyrimidines are different. So the purines are same, in which it is adenine and guanine, but the pyrimidines are different because the cytosine occurs in both DNA and RNA, but thymine occurs in DNA, and uracil occurs in RNA.
And we have an example of proteins being derived from mRNA, and the mRNA has been coded from the original DNA strand. And 3 letters equals to 1 codon. And then so we have AUG, which is a start codon, and then we have UGA, UAA, and UAG, which are stop codons, which go on the ends of the amino acid sequences.
So Kate, how is mRNA coded from DNA, which then a protein is later created in?
So through the process of transcription, as RNA polymerase moves down the template strand of DNA, mRNA is created. As the RNA polymerase moves down the template strand, adenine from mRNA pairs with the thymine of DNA. Guanine from mRNA pairs with the cytosine of DNA. The cytosine of mRNA pairs with the guanine of DNA, and the uracil of mRNA pairs with the adenine of DNA. And as we create mRNA from the original DNA template strand, the mRNA codons can then be created in the proteins. So Brown, as we have these codons from mRNA, how do we then translate it into proteins?
We translate the mRNA strand to a protein by using something called a protein codon chart. So how we use the protein codon chart is we have to know our mRNA codon first. So in our example, we have AUG as our mRNA codon. So how AUG will be used in the protein codon is we will start with our first nitrogen base, which is A. So we'll start in the middle, and then we'll just eventually get all the way out and find our protein. So to start this out, we start out with A. Then we go to U. We follow to U. And then lastly, we end with G. So our protein will code for MET. So I'll be talking about genetic mutations. And a mutation is a permanent change in the nucleotide sequence of a gene or another part of the DNA. So our first mutation I'll be undergoing is the silent mutation. And during the silent mutation, a nucleotide can change, but this will not affect the protein that's being created from the mRNA. So an example of this, we have the mRNA codon AAG, right? And that codes for the protein lysosome. And although our codon is changed to AAA and the nucleotide of G is replaced with A, our protein is still lysosome. So to sum the silent mutation up, although the nucleotide can change, this will not affect the actual protein that is being changed from the mRNA. Going to our next mutation, we have the deletion mutation. The name kind of says it itself. This removes a portion of the DNA sequence, and this changes how the rest of the code will be read by changing the total number of nucleotides. So for example, in our mRNA strand, one of our nucleotides is deleted or taken away from the sequence. This forces the rest of the strand to be coded in a different format.
So the third genetic mutation is insertion. The name kind of implies by saying nucleotides added to the second codon affecting shape and structure and how it works. So for our example, we have the mRNA strand, and we add a thymine to the second codon, and this is the resulting amino sequence after the insertion mutation. The last genetic mutation is point mutation. This is similar to silent mutation, except the proteins that come out from the mutation are not the same. So in point mutation, nucleotide is changed, overall resulting in a different amino acid added to the protein. So in our example, we have the mRNA sequence, and we changed adenine to thymine. And this resulted in a different protein from this codon, and results in this amino sequence.